from our ancient Sanskrit spiritual text, which explain the thoughts from Upanishad, and in the Vedic era, where women had equal opportunity to pursue their interests. Dear friends, it's my privilege to introduce today's speaker. Before introducing our guest, I would like to quote Yuval Noah Harari. I quote, unquote, we study science not to know the future only, but also to widen our horizons to understand that our present situation is neither natural nor inevitable, and that we consequently have many more possibilities before us than we can imagine. The same analogy we want to extend and expect from today's lecture. Dear participants, as you know, the Department of Physics of Dolphin Institute always brings to the intellectuals from across the globe for enriching our students' knowledge base in several ways. And in this continuation, today we have a cosmos personality from intellectual domain of this country. She is none other than Professor Sujata Sharma. Professor Sujata Sharma is an Indian structural biologist, biophysicist, and a professor at the Department of Biophysics of the All India Institute of Medical Sciences, Delhi. She is known for her studies in the fields of drug design and drug resistance of bacteria. Her studies have been documented by way a number of books, Warriors in White, an autobiographical account of some COVID-19 warriors at All India Institute of Medical Sciences, Delhi, and other leading hospital of India. The Department of Biotechnology of the Government of India awarded her the National Biosciences Award for the Career Development, one of the highest Indian science awards for her contribution to biosciences in 2011. She is also a recipient of Women Scientist Award of Biotech Research Society of India and the National Young Women Bioscientist Award of the Department of Biotechnology, which she received in 2006 and 2007 respectively. In 2020, she was awarded the Kalpana Chawla Excellence Award for her contribution in science. Really, she is foremost biophysicist in the scientific community of this country. Despite being very occupied, she accepted our invitation, shows her strong commitment for academia. So, without any further ado, I would like to invite Professor Sujata Sharma to deliver her talk on the integrity of science through the lens of biophysics. And further, I would like to request to our audience, if they have any questions regarding the subject, please go ahead and put them in the chat box. Please, madam, now mic is over to you.
Thank you so much, Dr. Ashish Rituri. It's a great honor for me to be present over here on this wonderful day of International Day of Girls and Women in Science, Technology, Engineering, and Medicine. And without wasting any more time, I think I will let my slides do the further talking. I'll share my screen.
Sure.
I hope my slides are visible to everyone.
Yes, ma'am. Yes, ma'am.
All right.
Because this
So
lecture
this,
is also broadcasting in our classrooms, so students are there,
okay.
researchers are there.
All right. Thank you. So this is the topic of my talk, the integrity of science through the lens of biophysics. And I'll introduce myself as Professor Sujata Sharma. Dr. Ashish has already given such a wonderful introduction. I don't even know if I deserve such great words, but thank you so much. And so let's start talking about biophysics. You know, it is a term that many people constantly wonder about. What is biophysics? So we'll start with the beginning in the 17th century. When Luigi Galvani, he was an Italian physician, he started doing research on muscle contraction and nerve impulses, and he found that electricity interacts with a biological unit called like muscle. So there is a connection between physics and biology, and his discovery led to the creation of lots of devices which we use even now, like ECG, EEG, and pacemaker. Those are common devices that you might have heard in the medical technology. They have all originated from biophysics. So after these initial observations were made, This term, biophysics, was introduced by Carl Pearson. He was an English mathematician. He introduced this term biophysics in his book called The Grammar of Science in 1892. And in that, he simply said that this is a science that links the physical and biological sciences. And at that time, he observed that this branch of science does not appear to have advanced very far, but he felt that it's going to have a very important future, and he was not wrong. So, what exactly is biophysics? Now that we have come this far, we know that it is an interdisciplinary science. So, nowadays, all sciences are interdisciplinary, they do not exist alone. And this science explains the laws and principles of physics which govern various biological processes. And it applies methods of mathematical analysis and computer modeling to understand, again, the functions of biological systems. And when it comes to the extent of what all it spans everything from molecular scale to whole organism. So from this, you must have understood that this is a very, very vast science, and it cannot be covered in a short time. If you just see that it starts from a small cell and it goes on to the whole organism, and it spans a lot of things. So I will limit my talk today to just a limited scale. If you see what are the branches of biophysics, there are many, but of course, four major branches are astrobiophysics, molecular, medical, and environmental. I have only listed four of them here, which are the major branches, and there are lots of sub branches of these branches as well. So when you wonder what is astrobiophysics, so it is an field of intersection between astrophysics and biophysics. It means that what we are doing is that we are trying to observe that what is the influence of astrophysical phenomenon upon life of planets. And these are two examples I've given. We could try to observe that what is the effect of supernovae, which are basically explosions in space, on life on Earth. That leads to a lot of climate change and winds, and basically that influences the biological processes as well. And of course, effects of cosmic rays which come out from the sun on Earth and life on Earth. The second branch is molecular biophysics. Now, that is what I have been working on since a long time, molecular biophysics as well as medical biophysics. So, molecular biophysics is a field that applies methods and concepts from physics, chemistry, engineering, mathematics to understand biomolecular systems. So, we not only study the molecular structure, but also the structural organization and what is a dynamic behavior, like do the molecules cluster together and perform a function, etc. And of course, the various levels of complexities from single molecules to supramolecular structures, viruses, and nowadays viruses are extremely, interesting things ever since COVID-19 has come. Then the third branch is environmental biophysics. Environmental biophysicists, they research the diverse microbial communities that inhabit this planet. And they also measure and model all aspects of environment from stratosphere to deep ocean vents. They also intend to track the pollutants, and in the end, it's about harnessing this knowledge into something that can help our future, like convert the biological molecules into biofuels, etc. Finally, medical biophysics, which is what we do at AIMS, science of physical phenomena occurring in biological systems, primarily in humans, in the normal state, and in diseases. So first we study biomarkers, that is an objective measure that captures what is happening in a cell or organism at a given moment. It could be a protein which is probably upregulated in a disease. And once we understand, okay, look, this is a biomarker, then what we do is that we solve the structure of that protein structures. Now this information is required to develop new drug targets for a disease. Then medical biophysics is also used in forensic biophysics, because we use biophysical techniques to understand forensic medicine. For example, if there is death and we want to know what has caused the death, then we need a lot of biophysics for that. And finally, imaging, as I had mentioned earlier, MRI, CT scans, EEG, ECG, all that is diagnostic imaging that also comes in medical biophysics. And these are some of the biophysical techniques like X-ray crystallography, spectroscopy, chromatography, and these are only few. I have not listed all of them because that would have been probably a lot. So now, just when you see these few slides, you will understand how much biophysics is surrounding you, and probably we are not even aware of how much biophysics that we are using in our daily life. Now, after giving a brief introduction of biophysics, I would like to show you two major examples that biophysics has helped us. Biophysics to the rescue in two pandemics in the last 50 years. Now, this slide shows you a number of pandemics that have taken place on the earth, starting from cholera, bubonic plague, Spanish flu, etc. But in the last 50 years, our earth has seen two major pandemics: AIDS, which started from 1981 to it's still going on. COVID-19 started in 2019. Now, in the coming few slides, we will see the role of biophysics, which helped in these two pandemics.
Excuse
First,
me. Are you changing the slides? Because
yeah,
you
I
are not.
am.
Yeah, here slides are not moving.
Okay, they're not moving.
It's,
I'm sorry. Uh, Shall I try to change the computer, or shall I?
sure. No, you can just slideshow in the same computer which you are using.
Just a minute. Presentation mode. Can you see them now? Can you? I'm changing them right now. Can you see them?
Uh, no, ma'am, no. Now you can again, please again, share it.
Okay, I'll reshare it. Can you see the change now?
Now it's working, right? Yeah.
Yeah. Okay.
Yes, ma'am.
So I'll just begin. Shall I go through the slideshow again quickly,
Yes, ma'am. Yes.
or
Yes.
shall I begin from now?
Now you can start from here.
Okay. So I was talking about the two pandemics that have taken place of in the last 50 years. Some were examples. Now, initially, you will see examples of cholera, bubonic plague, but I'm going to limit my talk only to AIDS and COVID-19. So I've changed the slide. Can you see the change in the slide?
This is the time span, I think, in your slide, biophysics to the rescue to pandemics in the last 50 years.
Yeah.
This is
So
the time
now,
span.
can you see the change in the slide?
Are you changing, ma'am?
Yeah, I'm changing. Can you
But
see?
we are not able to. No, yes. No, no. We are not able to see that.
Um, what I can try is I can join on another computer. Is that okay?
Sure, ma'am. Sure.
Okay.
Dear participants, sorry for interruption. So we'll back soon. Uh,
Are you able to see the slides?
Yes, ma'am, it is working. Now just please make a change.
Yeah, I'm making a change. Can you see it?
Yes, yes, yes.
Okay.
No, have you changed the slide, ma'am? Because
Yeah, I
we are
have.
able to see on the slide.
You're able to see the change now?
No, 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 ma'am.
Just a minute. Just a minute.
Sure, ma'am.
Share slides, and I'll have to upload the slides. Your computer, upload file. I'm uploading the file so that it's better.
Yes, it's okay. It will work.
कहाँ पे है ऐसे तो biophysics in the ground.
डेस्कटॉप पे जाओ
नहीं है. I'm not seeing it. ये रहा ना, तो फिर क्यों नहीं आ रहा?
ये कुछ और तरह से सही होगा पड़ा है
Share screen. Chrome tab. Window.
विंडो
Entire screen.
एंटर करके देख लो एंटर कर लो
तो entire screen. क्या?
शेयर नहीं हो
What is it?
शेयर का ऑप्शन नहीं आ रहा फिर से दोबारा शेयर करना
Slides.
नहीं ये उससे नहीं होगा यहाँ से देखो ये पावर पॉइंट लगा नहीं
Okay. I'm unable to do that. I don't know what to do.
Ma'am, can you send me the mail?
Yeah, I will do. I will do that. Yeah. So then it's possible.
Okay. Ma'am, are you able to see mail address at?
Yeah. I'm able to
Please
see
send on.
that. Yeah.
Yes, please send on.
I've sent.
Now we will share it by your end. Okay.
Okay.
Yes, ma'am. Ma'am, kindly unmute your mic.
Yeah. Okay.
Thank you, participants, for your patience, and sorry for this disturbance.
Yeah.
Yes, ma'am. Now continue.
Okay. So can we go to the next few
Yes, ma'am.
slides?
Yes.
Yeah.
Yes. Yes.
This is just the beginning. I was telling you about biophysics.
Yes.
The next slide. So again, maybe you can skip 3-4 slides, and we can come to the pandemics.
Next.
Next.
Hmm.
Yeah, but maybe 2 more. Yeah. Okay, next. All right, we can stop here. So basically, I was talking about the 2 pandemics in the last 50 years. First is AIDS, that is acquired immunodeficiency syndrome. So it was detected in June 1981, and the virus was human immunodeficiency virus, HIV. And so far, this pandemic has caused 36.3 million deaths. The next slide is going to show you. Can we have the next? Yeah. The second pandemic that is very recent, and we are very, very well aware of, COVID-19, that was detected in December 2019. And the virus was, of course, as everybody must be knowing, the audience, that it's SARS-CoV-2. And already it has caused 5.7 million deaths. Next slide. So this is the timeline of AIDS, how it all began. It began in May 1981 when the first reports came in California, and it was reported in New York Native newspaper. And then by June 1981, there were 5 cases in USA. July 1982, AIDS name was given because it was seen that that it hits the immunity. And then in May 1983, it was reported that there's a novel retrovirus, which is the cause. By June 1986, the HIV was named as the virus was named as HIV. The next slide will show how COVID-19 began. So on December 31, 2019, China informed WHO that there was 27 cases of pneumonia. And by January 11, 2020, China reported its first known death. And it was named COVID-19 on February 11, 2020, which is such a coincidence because now today it's February 11, 2022. So it's been exactly 2 years. And by March 11, WHO had declared COVID-19 as a pandemic. The next slide. So what I'm basically doing is I'm comparing the 2 diseases, both caused by viruses, both caused pandemics. So HIV origin was seen in Kinshasa, which is in Congo, and it was seen that it is zoonotic. Zoonotic means that the disease has come from an animal and it has jumped to humans. So it was back in 1920s, even though it was reported in 1980s, but it was back in 1920s. And before 1980s, probably AIDS was spreading, but we don't know because the number of cases were unknown. There were no reports. By 1981, by the time we all realized that there is such a disease, HIV had spread to 5 continents. Next slide. And this is the timeline of coronaviruses in 1931. Even though we hear about them now, but it was back in 1931. It was first detected in chickens, then in humans in 1965. And in 2003, we saw the first coronavirus pandemic, that was SARS-CoV. Then 2012, MERS-CoV, that is Middle East Respiratory Syndrome. And in 2019, as you all know, we had SARS-CoV-2. Next slide. So this again is a slide to show you that coronaviruses are also originating from bats. So they are also zoonotic, and they have some intermediate hosts like SARS-CoV had civets, and MERS-CoV had camels. And in case of SARS-CoV-2, the intermediate host is not yet known. There were reports of pangolins, but they were proved wrong. Now, in this slide, you will appreciate that SARS-CoV and MERS-CoV caused such few cases and very few deaths, 774, 885 deaths. But look at the extent of SARS-CoV-2. It has already infected 400 million people and 5.7 million deaths. Next slide. So basically, I've been talking about zoonotic diseases. So what is zoonosis? It's an infectious disease that has jumped from non-human animal to humans. And the pathogens could be bacterial, viral, or parasitic, and the spread is through direct contact or through food, water, or the environment. And it is amazing to realize that almost 60% of all human diseases and 75% of all infectious diseases are zoonotic. Next slide. So just this slide is just to show you how many zoonotic diseases are right now in the world from various countries. Next slide. So if you remember, I was talking about environmental biophysics in the beginning of this slide presentation. So this is where environmental biophysics comes in. If you see, these are the causes of zoonotic diseases: habitat fragmentation and deforestation, because what happens then is that the animals start moving towards human dwelling. So wherever you have human animal interaction, wherever you see more of it, there you will have zoonotic diseases. So intensive livestock farming, people have started doing that. So poor sanitation, unsafe disposal of animal waste has also caused zoonotic diseases. Unplanned urbanization, pollution, because when there's pollution, there's release of toxic compounds, and that leads to more mutations and horizontal gene transfer. Exhaustive agricultural practices, climate change, because when there's rise in temperature and there are floods and other such events, they lead to increase of pathogens. And bushmeat trading. Bushmeat trading means that these animals are being hunted. They're being hunted just to provide meat to humans. So the contact between human and animals again increases. And that leads to jumping of viruses from animals to humans. Next slide. So now from environmental biophysics, let's move on to molecular biophysics, because I've shown exactly how environmental biophysics is making us understand why this is happening. Now we have to find a cure. So we are moving to molecular biophysics. So if you see that both the viruses which caused AIDS and COVID-19 are RNA viruses, but there is a difference in the nature of viruses. In the case of HIV, it's a lentivirus, and in SARS-CoV-2, it's a coronavirus. And of course, there are structural differences, but the major difference that we should be concerned about is that there's a difference in the replication pattern within a host cell. And that completely changes how these pandemics spread. Next slide. So this is, I've just tried to put together how, what is the difference between HIV and SARS-CoV-2. I think there is, by mistake, I've written SARS-CoV-2 here, it's supposed to be here. So what SARS-CoV-2 is a simple virus. It attaches, if you see towards your left, attaches, it enters a cell, it replicates, assembles, and releases. But HIV is a retrovirus, which means that after entering, there are two more steps which don't happen in case of COVID-19. That is reverse transcription and genome integration. So that means your enzyme, an enzyme in this particular virus that changes RNA to DNA, and that particular DNA then incorporates itself into the host DNA, that is into the human DNA. And that is why HIV is so difficult to treat. Next slide. Of course, the modes of transmission in case of SARS-CoV-2, even though we fear SARS-CoV-2 much more than HIV because it spreads so easily, but actually this transmission is through only one way, that is mucous membrane of nose, eyes, and mouth, and of course air droplets, and only 1 to 4% deaths without treatment. How in case of HIV, it's due to unprotected sexual contact from mother to baby, blood transfusion, and if you don't treat, then there's more than 95% deaths. So it's much, much worse, HIV is. Next slide. Now coming to medical biophysics. I've already covered environmental and molecular. Let's go to medical biophysics. How can we treat a disease? Either we can prevent it or we can cure it. So next slide. So the question is that why don't we have a vaccine for HIV? That means why can't we prevent? You know, I was also surprised when I was working on this, that I realized that there is no vaccine for HIV, even though we have come out with the vaccine for SARS-CoV-2 within an year, literally. So the differences are these. First of all, as I had mentioned before, HIV genome integrates in the host cell genome. So if effectively it doesn't become a foreign body, it makes itself invisible to the host immune response. However, COVID-19, it doesn't integrate because it is not a reverse virus. It doesn't have reverse transcriptase. And then it is So that makes HIV not particularly immunogenic. And COVID-19 is, of course, very immunogenic. So humans mount a strong immune response to coronavirus. Even without a vaccine, there's a strong response in our body. And finally, mutations. HIV mutates very rapidly, much, much more than COVID-19 virus. Coronavirus have a significantly lower spontaneous mutation rate. Now, let me mention over here that RNA viruses mutate faster than DNA viruses, but even amongst them, there is a difference in the rates. And HIV mutates extremely fast. So within one human being, you'll have various types of, various variants of HIV virus. So it's very difficult to actually make a vaccine. Next slide. So two therapies for two pandemics. And these two therapies have been brought about by medical biophysics. In case of HIV, we have drugs. In case of COVID-19, we have vaccines. So the entire drug treatment of HIV is known as highly active antiretroviral therapy, HAART. And it completely covers the entire viral disease. You have nucleoside reverse transcriptase inhibitors in drugs, protease inhibitors, entry or fusion inhibitors, integrase inhibitors. In case of COVID-19, you have these kind of vaccines, inactivated subunit vector and mRNA vaccines. Next slide. So I want to just talk about how drug discovery was in pre-antibiotic era. In the earlier times, there was trial and error approach on naturally derived substances, and there was random screening. So how has biophysics completely transformed drug discovery? Next slide. So these are three scientists who transformed drug design. First was Roentgen, who discovered X-rays. Second was Max Loewe. He discovered diffraction of X-rays by crystals. And last is Alexander Fleming, who discovered the first antibiotic, and that is called the penicillin. All of them are Nobel laureates. And because of these three discoveries, the entire drug discovery system has changed. Next slide. So now what we do is that we use the protein structure, and we basically looking at the protein structure, we do the drug design. So we study the map of the protein implicated in the disease that leads to finally the drug design. So basically my lab is into three-dimensional structures of proteins that are implicated in disease. Next slide. So this is just to show you the various steps: protein isolation, purification, crystallization, and X-ray diffraction, and finally determining the protein structure, active site mapping, and drug design. Now, in case you're wondering if I'm wavering away from the two pandemics, I'm not. Next slide. I'm coming to AIDS, because the first drug for AIDS, which is still being given till today, was done by this particular technique. So this is the, if you see, this entire red surface is the protein, and the green area is the active site, and these blue residues are the active site residues. So we design a molecule that can go inside this active site and fit, so that this particular protein is not able to bind with its substrate. So in a way, it's a key and lock mechanism. Next slide. Now you'll have to go through the next 4 slides quickly. Now this particular is a protein, and this is the next slide. This is an active site. Next. So here your substrate binds. Next. So what we do is that we design a compound which can fit into the active site as well as good as a substrate, or maybe better. So once we bind this, next slide, the substrate is not able to come and bind. So that is, in short, how I've tried to do it in a very simplistic way so that students can understand how drug design is done, and that is how the drug was discovered for HIV. Next slide. So now you see, this shows you that a person who has HIV, who does not have HIV, a healthy person, his lifespan is 79 years. A person with HIV who's diagnosed at 20 years, but we give HAART, that is this particular drug template. If you see, the lifespan is almost equivalent to a normal person. However, if you do not give these particular drugs, it's as bad as just 32 years. So you can appreciate in the slide how much drug design through biophysics has helped AIDS patients. Next slide. So the drug target, like I was talking about drug target some time back, in this case was HIV protease. What HIV protease does is that it catalyzes a very, very important step. That is, it catalyzes the proteolytic cleavage of the polypeptide precursors into mature enzymes. And if this particular protease is inhibited, then AIDS is not able to spread. The virus is not able to uh, carry on its function. So that is why it is such an ideal drug target. Next slide. So this is the three-dimensional structure of HIV protease. It was done by Sir Tom Lundell, who's a structural biologist, and it is a homodimer. You can see that there is an active site, which is a catalytic cleft in the middle, and this is where your substrate binds. Next slide is going to show how elegantly the biophysicists were able to find a compound which bound to this particular active site, and they were able to inhibit this particular protease. And once they were able to do that, they could display that HIV stopped there and then in the person. Of course, the person has to continue having the drugs literally all his life. That cannot be helped. However, with the help of these drugs, the person's longevity can go on till almost 70, 71 years. Next slide. So this is just a surface diagrams. On the left is without the particular drug, and on your right is with the particular drugs, which shows you how it is getting inhibited. Next slide. Again, just to show how these compounds, which I have just shown in the structure, have made it to drugs, and they are being marketed as saconavir, indinavir. These are all the particular names of the molecules. From bench side to bedside, which is a dream of every medical biophysicist. So in these slides, I showed you how one pandemic was controlled. AIDS was controlled using drug design. Next slide. Now I'll come to COVID-19. In case of COVID-19, it has limited treatment options. We don't have drugs for it right now. In the above table, you can see that a lot of drugs were first tried out. Antibiotics, not recommended. Then ivermectin, hydroxychloroquine, plasma therapy, remdesivir, all of them were touted as the drugs for COVID-19 initially. Later on, after many studies, it was discovered that these drugs do not help. So by WHO, ICMR, it was not recommended. So what do we have? What options do we have as drugs? Absolutely nil. We can only depend upon oxygenation and steroids, and you know how dangerous excessive steroid therapy is. Dexamethasone is a steroid, because you might have seen or heard in the Delta second wave of when it came to India, that when steroid therapy was excessively done, you had other problems like mucormycosis. So it doesn't leave us with much options. So COVID-19 is an ideal candidate for vaccines. And luckily, fortunately, we were able to make vaccines for COVID-19 within 1 year. Now, how did this miracle take place? Well, it happened because, first of all, originally some research had already been done, because SARS-CoV and MERS-CoV, which are related to SARS-CoV-2, had already come, and some studies had been done already. Second thing that led to a quick response was, next slide, mRNA vaccines. Can I have that? Yeah. So I just want to show you how vaccines in the past were designed, just like, you know, I'd shown you how drugs were designed. So vaccines are basically biological preparations that provide active immunity. I'm sure you all know that. They contain an agent that resembles a microbe. Next slide. The goal of the vaccination is to develop herd immunity in the population. That means that majority of population is immune to a certain disease, so that would offer indirect protection to individuals who are not immune. So the desired vaccination for herd immunity in COVID-19 is at least 70%. So 70% of the population in the world needs to be, at least 70% needs to be vaccinated. And then you can say, okay, we've got herd immunity. Next slide. So the first vaccine was discovered by Edward Jenner, who is known as father of immunology. He discovered vaccine for smallpox in 1796. So what he had seen was that there were certain, smallpox was spreading all over, but one small set of population was immune to it, and those people were milkmaids, the one who used to work with cows. So that made him wonder, why are they not getting chicken pox and smallpox? So he realized that they already are sometimes infected from smallpox, which occurs in cows. So what he did was that there was a cow called Blossom, and he saw that this Blossom had infected a milkmaid known as Sarah Nelms. And so he took out the pus from the blisters from Sarah Nelms, and he injected into a child called James Phipps, who was basically a child of his gardener. And he was able to show that this child was immune to smallpox in the future. And so the terms vaccines and vaccinations derive from variole vaccinae, that is smallpox of the cow. And it wasn't that as if he immediately showed and it was taken up. He had to show it almost in 23 patients, including his 11-month-old son. Then only people believed him. Next slide. This is just artwork which showed how towards your left, you will see Edward Jenner trying to convince the milkmaid and saying that, could you please give me some of the pus which I can infect a child with. So the good news is, because of successful eradication efforts, smallpox vaccine was no longer recommended for use. So we have successfully eradicated. Next slide. So many vaccines after that came about, pertussis, diphtheria, polio, and now in 2006 we had hepatitis. All of them were being basically discovered using literally the same technology as Edward Jenner, killed virus, inactivated virus, and then of course some more technologies came about. But the really revolutionary technology was done in COVID-19, that is mRNA vaccines. Next slide. So now we have 4 types of COVID vaccines. The first, 3 of them were conventional, killed subunit viral vector, but mRNA, as I said, is the first time. Next slide. So these are some of the examples of COVID-19 vaccines. Inactivated vaccines means whatever I had shown just now, virus grown in culture and then killed to destroy disease-producing capacity. So we have Covaxin, CoronaVac, Covivac, and then there's an Iranian vaccine, which is Coviran, Barakat. And this is, of course, the duration, the age. Only in Covaxin's case, we have age above 15. The rest are all above 18. Then we have viral vector vaccines, in which vaccines use a viral vector to deliver genetic material. In our case, COVID-19, we are using an adenovirus. So we have Oxford AstraZeneca, and also in that we have Covishield, that is in India, Sputnik, Convidencia. Next slide. We have subunit vaccines. That means vaccines contain purified parts of the spike protein. Spike proteins are present on COVID-19 virus, that is SARS-CoV-2. So these are few examples. Finally, next. We have mRNA vaccines for the first time in the world, which are 2 of them, Pfizer and Moderna. If you notice, the efficacy of mRNA vaccines is much, much more as compared to other vaccines. We have almost 95 in case of Pfizer and 94 in case of Moderna. However, the other vaccines, if you had noted my previous slides, the efficacy was 78, 80. Even though the efficacy of anything above 50 is supposed to be a good vaccine, but of course, more the efficacy, the better the vaccine is. And only in case of Pfizer, we have age group above 5 years that can be safely given, and Moderna above 12 years. So it makes you wonder, why are these mRNA vaccines behaving or coming out with such better features as compared to the other vaccines? Next, I will just explain that quickly. Next slide. So basically, mRNA is messenger RNA. That's a single-stranded molecule of RNA that corresponds to genetic sequence, and it is read by a ribosome in process of synthesizing a protein. So mRNA will tell your cell, it will instruct your cell which proteins to make. So the idea of mRNA vaccines is that we create a synthetic mRNA in the lab. Now, this is also just like drug design. We are now designing a vaccine over here. We are not depending on natural processes. We are creating a synthetic mRNA vaccine which contain the spike protein sequence. So your mRNA, synthetic mRNA will be put into the vaccine, and that will be injected inside you. It will go inside your cell, and it will instruct your cell to manufacture the spike protein of the virus, which will be now seen as an antigen. Next slide. So this was, I just wanted to tell you how the technology developed. So what are the advantage of mRNA vaccines over traditional vaccines? First, safety. mRNA vaccines are non-infectious because we are not using your pathogen, we are not using your virus. We are simply synthesizing it in the lab. So there is a biosafety in the production facility, that the facility in which you're making your vaccine, you don't fear a outbreak of that particular disease. Secondly, clearing. The clearing of mRNA vaccine from the body can be anytime modified. It can be made faster, slower. It's all in your hands because you are working in the lab. Third is efficacy. It is very efficient in vivo delivery. And again, you use liposomes or nanoparticles to deliver them. This again, you can modulate, you can modify. Finally, production. That is the reason why we were able to get the Moderna and Pfizer so fast. Moderna designed this vaccine in literally 48 hours, that is 2 days. So there's a potential for rapid, inexpensive, and scalable manufacturing. Next slide. This is a slide to show you that total population is about 775 crores in the world, and we are already vaccinated for 484 crores. So 62.4% of world population is vaccinated, and our final goal is 70%. So we are this close to attaining herd immunity for COVID-19. So that is really good news. Next slide. This is my final slide. Now, since today is the International Day for Girls and Women in Science, so this is my role model, Marie Curie, who said that nothing in life is to be feared, it is only to be understood. It is also my favorite quote, and which helps me a lot whenever I'm feeling very low or I'm worrying about something. Nothing is to be feared, it is only to be understood. Now is the time to understand more so that we may fear less. So every time you fear anything, whether it is a scientific problem, whether it is an emotional problem in your life, I think the best way to deal with it is to try to understand. Write down the points, what is making you fear, how you can help the situation. Try to be as objective as possible. That is what I have learned from Marie Curie, whose autobiographies and biographies I've read quite a lot and which have helped me. So thank you very much for your patience, especially when there was a lot of technical glitch. I was really worrying that this presentation will not go through, and I'm so happy. Thanks to Dr. Ashish, who was so proactive and quickly getting the presentation from me and converting it and putting it down. I can't tell you how happy I'm feeling after giving this presentation. Thank you so much.
Thank you, ma'am. Really, it's a wonderful talk. And yes, we are very thankful to our audience and participants who make patience and they are well connected throughout your lectures. And sorry for any inconvenience because, but almost if end is good, everything is good.
Yeah.
And everything is smoothly is going in a hectic track as we are working in a digital area. Some clinches over there, but as you said that if end is good, everything is good.
Yeah.
So, ma'am, really, I'm very thankful on behalf of the Department of Physics and from the family of Dolphin that you spare your time and share your experiences with us and we are with our students. Ma'am, I think we have a little bit time, so I would like to ask one questions. Being a faculty of physics, many times I would like to integrate the different disciplines in our college, and definitely we are in the position to offer such kind of add-on courses. So can you suggest me what kind of add-on courses we can start in our college, which may be integrated through different discipline of sciences?
I think one, of course, is biophysics. Then you can even try bioinformatics because nowadays bioinformatics is a course which is really helping in case, for example, machine learning, big data. that is what is the future of medicine, and not just medicine but also molecular studies. So I think bioinformatics would be a really good choice.
Okay,
And
and one thing in mind, that's
yeah,
what about your insight on a quantitative biology?
yeah, of course, quantitative biology is also very important. It's something which has been in the history, and I think it will never go out of applications ever in the future. So yes, that would be a good idea too.
Okay, thank you, ma'am. And I think we have to sum up this whole session. And before uh, sum upping of all this session, as you definitely, as I know you from the social media, and I would like to share with our audience and participants that this is the one side of Professor Sujata Sharma, the flip side of her that she is a poet by heart. And I would like to conclude this whole session uh, with quoting some lines from her poem, Band Darwaje, I think, if I'm not wrong. Yes, ma'am?
Nice.
So
Yeah.
the beautiful lines, and really it will inspire the young girls and young researchers in this country. And really, I like those lines very much. And I quote, जितनी मोहब्बत तुमसे थी उतनी मोहब्बत अपने स्वाभिमान से है बंद कर लो सभी दरवाजे तुम मुझे कहाँ फुर्सत अपनी उड़ान से है and really I hope these lines inspire our students, especially girls. And Dolphin Institute always encourage the women and girls in sciences, and we have a lot of scholarship programs so they can join our programs, physics, biology, and other stream of the life sciences as well. So, dear participants, once again, a big thanks to all for being with us. And now we are signing off until we meet again. Thank you very much.
Thank you so much.
Thank you.